Hello everyone, Namaskar. Hope all of you are fine and happy because it is very important to be happy when you do something, isn't it? So let's begin with the part 3 of cell division and cell cycle. So in the part 1 we have discussed the interphase stage. In the part 2 we have discussed mitosis of the M phase of mitosis, and today we are going to study the M phase of meiosis. So let's begin. Now when we talk of meiosis, all of you know, the meiosis occurs where? Meiosis occurs in the germ cells, It occurs in the germ cells or reproductive cells. So for example, when there will be production of egg from ovary, we can say that the production of egg in ovary, it involves meiotic kind of cell division. Clear? Now, in meiosis, what happens? There is production of 4 daughter cells. In mitosis, what was the number of daughter cells produced from the parent cell? 2. Here it is 4. Production of 4 daughter cells, and they are half the number of chromosome as their parent. So imagine if the parent cell is twice n, what will be the ploidy of the daughter cells? The daughter cells will be how much? Only n. And if the parent is 4n, the daughter cells will be how much? Twice n. It becomes reduced to half. Clear? Then the third important point is the daughter cells are non-identical. Non-identical meaning they are not similar to one another. They are neither similar to one another nor they are similar to their parent. So this kind of cell division brings about what? It brings about variation, which is very, very necessary for adaptation and finally survival. Clear? Now let us see actually what will happen in the M phase of meiosis. So let me show you what will happen. So if this is the parent cell, it is diploid parent cell. When this cell decides that I have to undergo division, first it will enter interphase, isn't it? After that, it will enter the M phase. Now the interphase was common for both mitosis and meiosis, so I am not repeating it again and again. So imagine the cell has completed the interphase and enter the M phase. Now meiosis, when we talk about, suppose this cell, it enters meiosis, what will happen? First the cell will enter meiosis 1. Okay, so what happens when the cell will enter meiosis 1? It will produce 2 daughter cells. Here, and at the end of meiosis 1, both the daughter cells will be reduced into half the chromosome number. So actually when we talk about reductional division, we actually mean what? Meiosis 1. Meiosis 1 is actually where reduction occurs, reduction of chromosome number. Clear? Now when after meiosis 1, the cell will enter what? Meiosis 2. Now when the cell will enter meiosis 2, what happens? Definitely these 2 cells will enter meiosis 2. Now this cell will produce 2 daughter cells this cell will produce 2 daughter cells. Understood? So this is how actually the things happens. At the end of meiosis 1, we find 2 daughter cells which are half the number of chromosomes as their parent. After that, they enter meiosis 2, where these 2 daughter cells will again further divide. Clear? And as a result, we get how many total number of daughter cells? 4 number of daughter cells. I hope it is clear up till now. So let us first have a word diagram, schematic diagram. Meiosis, it is divided broadly into 2 major phases. One is what? Meiosis 1, and the second is what? Meiosis 2. Now the meiosis 1 is further divided into some important subphases. These are prophase 1, metaphase 1, anaphase 1, and at the end what we will find? Telophase 1. Here, I have kept a few space between prophase 1 and metaphase 1 because prophase 1 is very long and elaborate. In fact, it is the longest phase of meiosis, and it is further subdivided into 5 subphases. These are very, very important. The actual variation and recombination that we talk about meiosis, it actually happens during these subphases. So the first subphase is lactote. It is followed by zygote. Then comes pacitin. Pacitin is followed by diploid and finally diakinesis. Diakinesis. Okay. Now, once the cell completes meiosis 1, it enters another small gap phase which is called as interkinesis. Now, once it completes this gap phase of interkinesis, it will go and it will enter what? Meiosis 2. And the meiosis 2 is further subdivided into, I hope all of you can imagine what will be the names of these phases. These are prophase 2, metaphase 2, anaphase 2, and finally telophase 2. Okay, so as we already know that when a cell decides to divide, first it will enter what? Interphase. After that, it moves to the M phase. So a cell is there in interphase. After that, it moves to the what? M phase. And the M phase of meiosis involves this part. Clear? So if this is the cell which is entering the M phase, at the end of meiosis 1, we get what? This. This will be the resultant of the completion of this phase. After that, what will happen? The 2 daughter cells that have been produced these two daughter cells, they will further divide and produce a total of 4 daughter cells. This is what actually we are going to study in this lecture. Clear? Now if you talk about meiosis 2, you must have seen that what I have written: prophase 2, metaphase 2, anaphase 2, telophase 2. In fact, if you look into this diagram, don't you think it's a bit similar to mitosis? Yes, in fact, the physical process of meiosis 2 is similar to mitosis only. Whatever we have studied in mitosis, exactly those kind of physical processes will happen during this phase. So the actual reductional phase is which one? Meiosis 1. Now let us begin with meiosis 1. As I have already told you that the first step of M phase is what? Karyokinesis, which includes what? Division of the nuclei. And here the first phase is what? Prophase 1. Okay. Now what happens in prophase 1 is that the most elaborate and the complex physical things that happen, it actually happens during this phase. Take a sec. So the first subphase of prophase 1 was what? Lactotin. Let us see what will happen in lactotin. So imagine this cell which has completed interphase, okay? So if you remember nicely, at the end of interphase, what was the chromatin content? The chromatin content have become double. So it becomes how much? 4C. So this is a cell which has completed interphase. Now it is ready to enter the M phase, okay? Now the first part of the M phase is what? Meiosis 1. Under that we are studying prophase 1, and under prophase 1 we are studying the subphase lactotin. So let us see what will happen during lactotin. So when there is transition from interphase to lactotin, what we see? The chromatin, it will start condensing. The chromatin, it starts condensing and it will appear as chromosome, but they appear as very long and slender chromosome. The chromosomes, they appear very long and slender. So the chromosomes, they appear very long and slender, and gradually the centriole will start migration. It will start migrating towards the opposite pole. So this is the first phase of prophase 1, which is lactotin. So during lactotin, what happens? There is condensation of chromatin into chromosome. The chromosomes, they appear very long and slender, and there is generally migration of the centriole towards the opposite pole. Clear? Now, after lactotin, the next phase is what? Zygotin. Let's see what will happen during zygotin. So at zygote what happens? The chromosomes they further condense. Now they become more shorter, they become more thicker, they don't appear very long and slender as we saw them during lactogen stage. They'll be bit, bit more condensed. They appear a bit more short and thick. They appear like this. They appear shorter and thicker. Clear? The centriole will further move. So it keeps on moving until and unless it reaches the opposite pole. Clear? Meanwhile, we will see that there will be gradually disappearance of the nucleolus and other cell organelles like endoplasmic reticulum, Golgi apparatus, etc. So this is what zygote. Clear? This is very early zygote, the chromosome they have started condensing into shorter forms. Now gradually as we move towards the later part of zygote, we see that there will be pairing of the homologous chromosomes. Homologous chromosomes means, let us imagine that we have, as human beings, we are diploid organisms. How many chromosomes do we have? We have 46 chromosomes. Otherwise, we can also say that we have how many pairs of chromosomes? We have 23 pairs of chromosomes, isn't it? So, this 23 pair means from chromosome number 1, 2, 3 to chromosome number 23, it comes from where? Egg. Chromosome number 1, 2, 3, 4 up to 23, it comes from where? Sperm. Clear? And when both of them fuse to form a zygote, the ploidy becomes how much? 23 plus 23, it becomes 46. That is why we call it as twice n equals to 46, isn't it? Now, this chromosome number 1 from the egg, this chromosome number 1 from the sperm, they are morphologically same, they carry the same genes. Clear? That means if chromosome number 1, it is carrying genes for, suppose let us talk about height. Okay? So chromosome number of egg will also carry the gene for height. Chromosome number 1 of sperm will also carry the gene of height. So that is why they are called as what? They are called as homologous chromosomes. Homo means what? Same. Morphologically same, and they also carry the same genes. Clear? Now in this case, if you see, you can notice this very visibly. This one is similar with which one? This one. So this will pair with this. Clear? And if you see, this one is similar with which one? This one, morphologically, isn't it? So we will see that if I mark it as chromosome number 1, this will be the another one. If I mark this as chromosome number 2, this will be the another one. So 1 will pair with 1, 2 will pair with 2. And this process of pairing up of homologous chromosome, it is called as synapsis. So I am drawing here. So this chromosome pairs with the same chromosome, morphologically same chromosome. And the pairing of these two homologous chromosomes is called as what? Synapsis. Okay? This leads to the formation of what? This leads to the formation of tetrads. If you see, this is one chromatid, this is another chromatid, this is the third number chromatid, this is the fourth number chromatid. How many chromatids are there? 4. So it is called as what? Tetrad. Or if you see how many chromosomes are there together? 2. So it is also called as formation of bivalence. Clear? So during zygote, what happens? There is pairing up of homologous chromosomes which leads to the formation of what? Tetrads or bivalence. This is very very important. We have seen that earlier in NEET there has been question from this part of the syllabus. So I think meiosis 1 is a bit complex phenomena. You have to be very very attentive, but it's very interesting too. You get to know a lot of stuff. You get to know why variation occurs. You get to know how variation occurs. Actually we are moving to that phase where we will know how the variation occurs. This is the beginning. First there will be what? Pairing up of the homologous chromosomes, and this process is called as synapsis. Now imagine these two chromosomes, chromosome number 1, clear? So we have one chromosome from mother, one chromosome from father, isn't it? The homologs come together, so they might get separated away, isn't it? So there must be some kind of interaction or adhesion between them so that they do not get separate. For that there is formation of what? For that there is formation of synaptonemal complex. There is production of synaptonemal complex. This keeps both the homologous chromosomes together. You can think this is some kind of adhesive or glue, basically bonded by some kind of protein molecules. Clear? So this synaptonemal complex formation is very important because it will facilitate crossing over in the next phase, that is Peckytin. So I hope you have understood what is happening in this phase. Now let us move to the next phase, which is Peckytin. So in Peckytin what will happen? Gradually the movement is increasing. Now what we see, the homologous pair that we had seen, there was formation of homologous pair entering zygote. There will be crossing over. So there is crossing over between the non-sister chromatids of a homologous pair. If you remember, I had told you earlier in the earlier lecture, the two chromatids that are connected at the region of centromere, these two are called as what? These two are known as sister chromatids. These two are called as sister chromatids. Clear? Now the homologous pair will cross over. Now these two red structures, these two red chromatids, they are sister. These two green chromatids, they are sister. But this red and this green chromatids, the one from the maternal side and the other from the paternal side, these two are called as what? Non-sister, because they are not the part of the same chromosome. So they are non-sister chromatids. So in Peckytin what happens? There is exchange of segments. There is exchange of segments between the non-sister chromatids of a homologous pair, and this exchange is called as what? This exchange is called as crossing over. This is very, very, very important. There has been question in NEET, previous NEET, where they ask questions like crossing over is found in which phase of meiosis. They are not going to give you options like prophase 1. Usually they don't give. they will give you options like lactotin, zygotin, pectin, diplotin. So the answer will be what? The answer is pectin. Now at the site of the crossover, you know, where the non-sister chromatids they cross over, there is formation of what? There is formation of a recombination nodule. There is formation of a recombination nodule, and this process of crossing over, it is aided by an enzyme which is called as enzyme recombinase. So this enzyme recombinase, it helps in the process of crossing over. Clear? And we see that there is exchange of segments between non-sister chromatids of the homologous pair. So this is what happens during pectin phase. Now let us move to the next phase, which is diplotin. So in diplotin what happens? During diplotin there is dissolution of the synaptonemal complex. Dissolution means naikya hoi jabo, okay? So is a complex eta duita ke ekelage bandhi rakhise boli koisilu, jabo dia nai. So what happens? Diplotinot ki hobo? Hey jitu force asile ba attraction asile, it will dissolve, naikya hoi jabo. So then what will happen? Duita konobai bandhi rakhise, laheke jitu gath asile jule bandhi rakhise naikya hoi gol. Duita tani bono opposite phale, because they will want to move apart from one another. Eneke asile, they will try to move apart from one another when they will try to stretch oppositely, isn't it? So যেতিয়া এনেকে দুইটা opposite ফালে টানিব try কৰিব, what we will see, a capital X-shaped structure is formed. Is a capital X or is a structure formed. This structure is called as chiasmata. Here, this is very important. During diplotin, what happens? The first step is what? Dissolution of synaptonemal complex. So what will happen? You will see this kind of a structure. More bahirot kyo aaki disu so that it's a bit more enlarged drawing. It's more visible for you. This structure is called as what? The X-shaped structure is known as what? It is called as chiasmata. Clear? So this is very, very important during diplotin step. One is what? Dissolution of synaptonemal complex, and the second part is what? Formation of chiasmata. Now it is found that in oocytes of certain vertebrates, oocytes means जो egg produce হয় তেনেকুৱা cellক আমি oocytes বুলি কওঁ. In oocytes of certain vertebrates, the diploid stage may be prolonged. মানে দীঘলীয়া হ'ব পাৰে. কেতিয়াবা ইমান দীঘল হ'ব পাৰে যে সেই ষ্টেজটোতে it can remain for a few months or years. Okay? তেনেকুৱা এটা diploid stage of কি বুলি কয়? Such a prolonged diploid stage is called as dictyotin stage. Or a dictyotin stage to quote by, it is found in oocytes of certain vertebrates. It is found in oocytes of certain vertebrates. So please comment if you know any vertebrates where you find the dictyotin stage here. So now let's move to the final stage of prophase 1, which is what? Diakinesis. So in the meantime, we will see what? The centriole has already reached the opposite pole. Clear? Now, what happens in the last stage, in the stage of diakinesis? There is terminalization of chiasmata. What happens? If you remember, with the homologous chromosomes, the non-sister chromatids, they were attached nowhere at the point of chiasmata. As I stated, because of the dissolution of the synaptonemal complex, the homologous pair, they try to move away from one another. So gradually what happens? The chiasmata slips. That is called as terminalization of chiasmata. So what we will see? Okay, now the homologous chromosomes, they will appear this way. This is one homologous pair, this is one homologous pair, and if you observe nicely, you can see that they have exchanged their segments, you know, there has been crossover. Very visible. This is how the homologous chromosomes they appear now at the stage of diakinesis. Clear? Now what will happen? There will be disintegration of the nuclear envelope at the end of diakinesis. And I have already told you in the lecture 1 when we were studying mitosis that when this nuclear envelope breaks down, it is an indication that we have to enter what? The cell has to enter metaphase. So the disintegration of the nuclear envelope marks the beginning of metaphase 1. Clear? Moreover, what we will see now? Any guesses? There will be initiation of the assembly of tubular proteins because during metaphase, what will happen? Spindle fibers must be produced. Clear? So this is the end of very prolonged, very interesting prophase 1. It is the longest phase of meiosis. Happen in metaphase 1. So when the nuclear membrane was disintegrated, what will happen? The chromosomes, they will spread in the cytoplasm. Now during metaphase 1, what will happen? The homologous pairs of chromosome, they will come and align themselves at the equator of the cell. Clear? And this alignment, it is very random. So we had this pair, isn't it? So the homologous pairs, they come and align themselves randomly on the equatorial plane, and there is formation of what? Spindle fibers, and the spindle fibers, they attach to the centromere of the chromosomes. Clear? Now during mitosis, I had said that it led to the formation of metaphasic plate. Now because now we have bivalent or tetrads or pair which are found at the equator of the cell, we call these as double metaphasic plate. So this is what happens during metaphase 1 of meiosis 1. The chromosomes, the homologous pair, they come and align themselves at the equator of the cell. Now, what will happen? After this, the cell will enter what? Anaphase. Anaphase 1. So during anaphase, what will happen? During anaphase, what happens? The sister chromat, the homologous pair, they separate. Okay? There will be shortening of the spindle fibers, and the homologous pair, they move to the opposite pair, So after metaphase, there will be transition to what? Anaphase 1. So during anaphase 1, what will happen? During anaphase 1, these homologous chromosomes, they separate. This chromosome will then move to this pole, this chromosome will move to this pole. But remember one thing, during mitosis, we have stated that there is splitting of centromere. Have you seen here splitting of centromere? No. Only there is separation of the homologous pair. So what we will see here? So there is separation of the homologous pair. You have to remember this point, it is very important. There is no splitting. There is no splitting of centromere. Often we see that in NEET, they give you questions on cell cycle and cell division. where they give you 2 columns and they ask you to match the following columns, you know. So you have to be very precisely remembering this, that in anaphase 1 there is no splitting of centromere. Now after anaphase 1, what will happen? The cell will move to telophase 1. So let's see what will happen. The chromosomes will reach their respective poles. Gradually there will be formation of the nuclear envelope and formation of daughter nuclei. So on this pole, this kind of a daughter nuclei will be formed, and on this pole so there is formation of 2 daughter nuclei at the end of telophase 1. Clear? And if you observe nicely, you can see that we started with how many chromosomes? 4, isn't it? Now we have how many chromosomes in one nuclei? 2 and 2. They have been reduced. It has become how much? N. So at the end of telophase 1, what we will see? There will be production of 2 haploid daughter nuclei. Now, after this, there can be a small interphasic stage, and it can transit to the next phase, that is meiosis 2, or there can be cytokinesis to produce 2 daughter cells. After that, there can be interkinesis, and those 2 daughters will next move to meiosis 2. So I am doing the second one, where I will show you that after cytokinesis how 2 daughter cells will be produced, haploid daughter cells will be produced, and how further those 2 haploid daughter cells, they will enter the meiosis 2 phase. Clear? So now, as we already know, it is a animal cell. What will happen in cytokinesis? There will be formation of furrow, isn't it? So by formation of a furrow, what will happen? There will be cytokinesis and production of 2 daughter cells. So in this way, what we will find? Production of 2 daughter cells. So you must have already observed variations. So at the end of meiosis 1, we have found 2 haploid daughter cells. These are the 2 haploid daughter cells. Now these 2 haploid daughter cells, they can enter meiosis 2. So as I have already stated earlier, the meiosis 2 is physically, in terms of the various steps, it is similar to mitosis only. So let's see what will happen. So prior to meiosis, there was a small gap phase, what shall we call this one? Interkinesis, isn't it? So during this interkinesis or the gap phase, what happens? The cell may synthesize necessary proteins like tubulin, but no DNA replication occurs. DNA replication occurs only once, that was during the interphase stage that we have studied in part 1. Lecture 1 we studied that during interphase stage, during the S phase of interphase, there was replication of DNA. So that is the only one time when DNA replication occurs. So don't get confused that because we have 2 meiosis, there will be twice replication. No, DNA replication will occur only once. Clear? During this interkinesis, the cell, if it feels necessary, it might synthesize tubulin proteins or other necessary molecules, but no DNA replication. Clear? Now let's see what will happen in meiosis 2. So meiosis 2 is similar to what? Similar to mitosis. So actually what happens at the end of meiosis 1, we see that during telophase there is formation of 2 daughter nuclei. Once the nuclear envelope is formed, the chromosomes, they might become slightly long and slender. They decondense a bit, but they do not decondense entirely to the stage of chromatin. So during meiosis 2, what happens? Meiosis 2, the first phase of meiosis would be what? Prophase 2. So during prophase 2, what will happen? The chromosomes, they will start condensing. So I am showing here only. You have to write down all the steps properly, you have to draw all the diagrams properly. I have the liberty of using a board where I can rub and do the things in the same diagram. So in prophase 2, what will happen? These chromosomes, they will become more condensed. Okay, they will show condensation. They will show further condensation. So I have drawn here. I am not repeating the same thing in the other cell. So they will be a bit more condensed. Okay. Moreover, the centriole will migrate and come to the opposite pole. Clear? And by the end of prophase 2, what will happen? The nuclear envelope will disintegrate. So we have seen that the centriole has migrated to the opposite pole and there is disintegration of the nuclear envelope. And the breakdown of nuclear envelope marks what? The breakdown of nuclear envelope marks the beginning of metaphase. So now we will see what will happen in metaphase 2. So in metaphase 2, what happens? The chromosomes that had been spread in the cytoplasm, they come and align themselves with the equator of the cell. Now this alignment is also very, very random, not following any definite sequence, okay? So I am going a bit fast here because this is very similar to you. Already it is done in the lecture 2, mitosis. So they come and align themselves at the equator of the cell. So this is what happens during metaphase 2. If you see, it is physically similar to mitosis. There is formation of what? Metaphasic plate. So this is the metaphasic plate, this is the metaphasic plate. Now we move to anaphase 2. So in anaphase 2, what will happen? There will be splitting of the centromere, okay, and separation of sister chromatids. So this is exactly similar to mitosis. So you have to remember one thing: in mitosis anaphase and anaphase 2, there is splitting of centromere, but in anaphase 1, there is no splitting of centromere. Can you tell me in the comment section what is the shape of this chromosome? Also the shape of this chromosome. Okay, this is how anaphase 2 will look like. There is separation of sister chromatids. Now let us come to the last phase, which is telophase 2. So what will happen after anaphase? These two chromosomes will reach the opposite pole, these two chromosomes will reach the opposite pole. So I am making the diagram here only. Let me rub this. So if you see, at the end of telophase 2, what we find? The production of haploid daughter nuclei. And when this haploid daughter nuclei be produced, the cell will enter the next phase, which is cytokinesis. So from this cell, if cytokinesis occurs, we will get 2 daughter cells. From this cell, we will get 2 daughter cells, isn't it? So at the end of meiosis 2, what we find, there is production of 4 daughter cells. And if you observe closely, you will find that all the 4 daughter cells are not identical to each other. Just have a look. Here the chromosome number 1 and here the chromosome number 1. Are they similar? No. If you see the chromosome number 2 here and the chromosome number 2 here, are they similar? Similarly, if you compare both of this, are they similar? If you compare this and this, no. This brings about variation. That is why meiosis is so very important, because it brings about variation. And variation actually helps in adaptation, which is necessary for survival. So if you remember, we took a mother cell. The mother cell entered what? Meiosis 1. After that it produced how many daughter cells? 2 daughter cells. Clear? Those 2 daughter cells entered meiosis 2. And finally after meiosis 2, how many daughter cells were produced? 4. So these are the 4 haploid daughter cells. So generally in case of animals, these are like, this can be gametes, this can be eggs or sperms, isn't it? So this is the end of this lecture. I hope you enjoyed it, and you have any kind of feedback, do let us know in the comment section. So I'll give a short snippets of the entire chapter, cell division and cell cycle, in another video. And there are certain other things like, we have to know the significance of mitosis and meiosis. Have a look in the book of NCERT and do let me know if you find it. Thank you. Bye bye. See you in the next video.